Hi guys, I just wanted to go over the card sort with you just so you knew that you had the right order. And it's on today's assignment too. And so it kind of double, you can go through it with me and then go fix it on your assignment today. So I'm just going to kind of tell you what you're seeing as you go through these two. So this was card one. So in this process, we're taking one cell and we're going to two cells, right? This cell is replicating. And so this is the first card. I'm going to go ahead and show you the last card. This is the last card, okay? And so we're taking one cell and making two. In this picture, you can see the nucleus is still intact, okay? And I'm going to go ahead and go through the phases. This is actually interphase when you look at your labels. And this cell would just be doing what it normally would do. Whatever its function is, it would be doing its function, okay? Picture two, we're seeing the nucleus has dissolved, the nuclear membrane has dissolved, and we're seeing DNA coiling into chromosomes. DNA is super long and skinny, and so you're actually, if you can see it, it means it's really coiling up. And so this would be the beginning of prophase, okay? And prophase is actually going to be cards 2 through 5. So this is the beginning of prophase. Here is the third card, and we can actually see the chromosomes. So the chromosomes have completely coiled. So these little butterfly-looking guys, these are the chromosomes. And you might want to label it if you've printed this out. Number 4, we see this kind of dotted line beginning to form. That's the spindle. This is a group of proteins that are going to move these chromosomes. And so the spindle has begun to form in 4. This is still prophase. And in 5, the spindle has completely formed, and you can see it attached to those chromosomes. And so 2 through 5, that would be prophase of this process. Card 6, those chromosomes have lined up along the middle of the cell. If this was a human cell, you would have 46 of these lined up, okay? And you can see the spindle is what has moved them. They've brought them to this midplate. And guys, card 6, that's metaphase. Metaphase, middle. They've lined up on the middle. And it's only this card. Metaphase is when they're lined up, and that's it, okay? Card 7, we see that something's happened to our chromosomes. And in card 7, those chromosomes have divided, okay? And they're moving to opposite ends of the pole. This is the beginning of anaphase. Anaphase away. Now, guys, the reason this can happen is because these are copied chromosomes, okay? And so here, I said there'd be 46 in a human, and there would, but you would have 46 that can go this way when they split and 46 that can go this way when they split. And so in card 7, we're seeing them beginning to divide. So that's anaphase. Card 8, they're a little farther. Card 9, they're a little farther. This is all anaphase. We go down to our double cards. We find card 10, and we see that the cell is beginning to elongate. So you can see it's starting to look like two cells, and those chromosomes are even farther apart. This is still anaphase. Anaphase is long, okay? 11, they're farther apart. It's getting, the spindle's kind of getting skinnier, still anaphase. 12, they're really on opposite parts. We almost have two cells. We still have a spindle. This would be anaphase as well. Card 13, notice it looks a little different because our spindle has disappeared. Okay, so this is the beginning of telophase. Telophase, because our spindle is disappearing, and that's card 13. And then in card 14, our chromosomes begin to uncoil. That's part of telophase as well. And we almost have two cells. So 13 and 14 would be telophase. And then card 15, the cytoplasm has completely divided. So this is cytokinesis, and we have two cells. Okay, so we've gone from one cell to two cells. You've heard the phases: interphase, prophase, metaphase, anaphase, telophase, cytokinesis. I hope that helps. Reach out if you have any questions for me, and I hope you're staying warm today and safe inside.